So I'm Patrícia Inácio, Bionews Service correspondent for Friedrich's Ataxia News. Dr. Puccio, thank you for being part of Friedrich's Ataxia News official coverage of the International Ataxia Research Conference here in Pisa, Italy, and for agreeing to do this filmed and recorded interview. So our first question is, which particular sensory impairments seen in patients with Friedrich's ataxia were mimicked in the new mouse model?
So, thank you for interviewing me already. So, indeed, we generated a new mouse model that is mimicking the proprioceptive impairment. So, this is the impairment that gives you the position of your body. So, this is one of the first impairment that is affected in FA patients. And so, it's in the dorsal root ganglion. That's particularly what we have modelized in the new mouse model. And so, that gives us sensory ataxia.
You previously demonstrated in a Friedrich's ataxia mouse model that gene therapy can correct heart abnormalities. Now, the same approach corrected sensory ataxia. Given that cardiomyocytes and proprioceptive neurons are the most affected tissues in the disease, how important is the discovery that this gene therapy can be successfully applied to both types of tissues?
So, indeed, we had previously shown that cardiomyocytes were able to not only be prevented from degenerating, but actually we can reverse them. So that was our first surprise, and that was our first discovery. But we did not know if for the neurons we could do the same thing, because what happens is when the neuron degenerates or becoming dysfunction, the axons, so the big long parts of the neurons, actually retract. And so whether we could correct that and correct the neurons and actually then also get the neurons to regenerate was a question. So this is actually what we're able to show, that we can prevent neuron differentiation or neuron degeneration, but we could also rescue neurons that have already been degenerated.
So the treatment prevented nerve cell death in the dorsal root ganglia, as you were saying, of the animals. Why is it important to preserve these neurons in particular?
Because, as I said in the beginning, these are the primary neurons that are affected in FA. Not the only neurons, but they're primarily affected. There's also neurons in the dentate nucleus. But these are the neurons that give the position of the body. And these are the neurons that help get the right equilibrium and be able to be stabilized. And so if we can preserve these neurons or preserve them and even not regenerate the neurons, but regenerate the axons. But if we can preserve these neurons, then we should be able to preserve the ataxic phenotype.
When a gene therapy combo was applied at a more advanced stage of the disease in mice with key disease symptoms, positive results were also obtained. Could you briefly explain which key symptoms these were and the extent of their improvement?
Okay, so that's I was mixing the two together before, but it's okay. So when we treated the animals pre-symptomatically, so I'll just come back on. When we treated the animals pre-symptomatically, we prevented the degeneration. We then treated the animals post-symptomatically, so they had already an ataxic phenotype. They already had a loss of sensory phenotype, which is what the proprioception is. And we can measure this by EMG, the same way it's measured in clinic. So when we treated the mouse post-symptomatically, we were able to reverse the ataxic phenotype, so they became less ataxic. Reverse the loss of the sensory wave, which is measured by the EMG, and actually extend the lifespan of the animal also, and have an animal that actually had not such a severe phenotype. We weren't able to correct completely everything because there's we have a cerebellar phenotype that we were able to partially correct only.
So the results provide a proof of concept for developing gene therapy for Friedrich's ataxia. What is the next step for this project? Can one envision tests in patients in the near future? Are you optimistic that a therapy that worked in a mouse model might be translated successfully and safely to humans?
So the next step is exactly to try to see how we can translate this into humans. So there's already quite a few steps of trying to identify the right vector. So we've previously shown that too much frataxin is actually not good. So, but the different labs and the companies developing gene therapy approaches for FA know this. And so I think most companies and most labs are developing vectors that are expressing low levels of frataxin. And then, so that's the first step, to have some more clinical grade vectors, and then test a dose response to see how low can we go. And so here, this was a kind of a proof of concept with a high level of frataxin, but then we need to go on the dose response and see how low we can go. And at the same time, there needs to be the development of the clinical grade vector and toxicity studies and large animal studies. So, but yeah, I'm confident that there will be something in the near future. There's enough companies working on it, so to do something, yeah.
So the therapy is injected intravenously, which means into the blood circulation. How specific it is to the targeted tissues? Were any concerns seen in the mouse model that the therapy affected tissues other than the target tissues?
So it's actually it's not a specific targeting. So the vector we're using is targeting quite well the dorsal root ganglia neurons, the heart, but it's also targeting the liver. It's also targeting muscles. The frataxin gene is expressed everywhere, and so if we don't have too much expression, targeting every single tissue is not a problem. So that to me is not a concern. Actually, the more tissue we target, the better it is. It's just then the level of expression that's going to be difficult. So for now, I don't envision if we're at normal or endogenous level or just regular level of expression, I think we're fine with targeting multiple tissues because frataxin is expressed in all tissues.
So just to conclude, is there anything else you would like to discuss or point you would like to further clarify?
No, I think this is exciting. I think that gene therapy is something very promising. I think that it's important to continue moving forward, but I also think that it's important to continue moving forward on pharmacological approaches also, because we don't know if gene therapy will be able to hit all the cells that need to be hit. So that's something important. So I think that it might be a combination of a gene therapy and a pharmacological therapy that will be used. So I think both approaches need to be continued to be developed.
Okay, thank you so much.
You're welcome.